Imagine spending 5 days in a room with a piece of linen that quietly dismantles everything you thought you knew about history, science, and the boundaries of human understanding. That is exactly what happened to Barrie Schwortz in October of 1978. He walked into the Cathedral of St. John the Baptist in Turin, Italy, as a professional photographer with a job to do. He walked out 46 years later, still trying to explain what he saw. And now, in 2024, the latest round of DNA analysis conducted on material collected from the cloth surface has produced results that have left even the most seasoned researchers struggling for words. Not because the findings confirm what believers hoped for, but because they do not fit any category, religious or scientific, that we currently have available to us. Barrie Schwortz was not someone you would expect to become the world's leading documentarian of Christianity's most debated relic. He was born and raised in a Jewish family in Los Angeles. He had no theological investment in Christian artifacts, no childhood fascination with church history, and no particular reason to care whether a strip of ancient linen in northern Italy was genuine or not. What he did have was an exceptional eye, decades of experience in scientific and technical photography, and a reputation for precision that made him the obvious choice when the Shroud of Turin research project began assembling its team in the late 1970s. The project, known as STURP, brought together more than 30 specialists from disciplines including physics, chemistry, textile analysis, and forensic science. Their goal was straightforward: apply rigorous scientific methodology to one of the most controversial objects in the world and document everything they found. Schwartz's role within that team was to serve as the official documenting photographer. He was not there to form opinions. He was not there to advocate for any position. His job was to create an accurate, comprehensive visual record of every test, every sample location, and every observation made during the 5 days and nights the team had access to the cloth. It sounded simple enough. What he discovered during those 5 days was anything but simple. The cloth itself measures roughly 14 feet in length and a little under 4 feet in width. It bears a faint, sepia-toned image of a human body, front and back, as though a person had been laid on one end of the cloth and the other end had been folded over them. From a distance, the image appears almost ghostly, barely visible against the aged linen. But when Schwartz began photographing it under controlled conditions with different wavelengths of light, the strangeness of what he was looking at began to assert itself. The first thing that struck him was the complete absence of any paint, dye, or applied pigment in the image areas. As someone who had spent years photographing artworks, manuscripts, and archaeological materials, Schwartz knew what applied artistic media looked like under magnification and under varying light conditions. Paint leaves layers. Dye penetrates fabric through capillary action. Even the most skilled medieval artists, working with the finest materials, leave traces of their technique when examined closely enough. The shroud image left none of those traces. Under magnification, the image appeared to be the result of a chemical change confined to the outermost surface of the topmost linen fibers, penetrating no deeper than a few hundred nanometers. The underlying threads showed no discoloration. The reverse side of the cloth was clean. Whatever had created this image had done so with a precision and a superficiality that no known painting or dyeing method could reproduce. Then came the ultraviolet photography. Aged linen fluoresces under ultraviolet light. The breakdown of organic compounds in old cloth produces a faint glow that is entirely predictable and well understood. The background linen of the shroud behaved exactly as expected, producing the characteristic fluorescent response. But the image areas, the portions of the cloth that displayed the body figure, did not fluoresce. They appeared darker under ultraviolet light, not lighter. This told Schwartz and the chemists working alongside him that the fibers in the image region had undergone a different kind of chemical change, one that had altered their optical properties in a way that is inconsistent with any conventional artistic process and inconsistent with natural aging. But nothing prepared Schwartz for what happened when the STURP team ran the shroud images through a VP8 image analyzer. a device originally developed for NASA to extract three-dimensional topographical data from photographs of planetary surfaces. When a standard photograph is processed through this device, the result is distorted and incoherent because photographs encode visual light and shadow, not actual distance measurements. When a shroud image was processed through the same device, it produced a coherent, accurate three-dimensional representation of a human body. The tonal variations in the image corresponded not to how a painter would render light and shadow, but to the actual distance between the cloth surface and the underlying body. Areas of the face that would have been closest to the cloth, the tip of the nose, the brow, the cheekbones, appeared darker. Areas that would have been farther from the cloth appeared lighter. The image contained real spatial data. It was, in the language of the researchers, distance mapped. No painting from any era does this. No known artistic technique produces this property. Schwortz also documented the photographic negative quality of the image. When he processed his film and examined the negatives, the body image appeared as a positive. This meant the shroud image itself was already in negative, with light and dark values reversed relative to how the human eye perceives a real object. The nose appears dark on the cloth. The eye sockets appear lighter. It is only when the image is photographically reversed, turned into a negative of a negative, that a natural and detailed portrait of a human face emerges with clarity. Photography as a technology did not exist until the 1820s. The medieval dating of the cloth, if accurate, would place its creation approximately 5 centuries before anyone understood what a photographic negative was, let alone how to deliberately encode one into a piece of linen. Sturp concluded its 1978 examination with a carefully worded summary. The team confirmed that the image was not the product of any artistic technique they could identify. They confirmed the presence of blood and serum proteins in the stain areas. They stated that the image appeared to represent a real human body bearing wounds consistent with crucifixion as described in first-century Roman sources. What they could not determine was how the image had been formed. That question remained open when the team dispersed, and it has remained open ever since. The carbon dating conducted in 1988 complicated the picture considerably. Samples taken from a corner of the cloth were sent to 3 independent laboratories, each of which returned a date range placing the linen's origin somewhere between 1260 and 1390 AD. The announcement was treated in much of the press as a final verdict. The shroud was medieval. The matter was closed. Schwortz, however, spent years examining the methodology of that dating process and found reasons for serious concern. The samples were drawn from a single corner of the cloth, a region that had sustained damage during a 1532 fire, had been handled more extensively than any other part of the cloth over its documented history, and showed signs under examination that it may have included repair threads introduced at a later date. The most contaminated, most structurally compromised region of the entire cloth was the region from which the dating samples were drawn. Schwortz's position, shared by a number of textile experts and statisticians who later analyzed the published data, was not that the 1988 dating was fraudulent, but that it was methodologically inadequate to answer the question it was supposed to answer. Dating one corner of a cloth does not date the cloth. For the next several decades, Schwortz continued his research independently, maintaining what became the primary online repository of shroud science through his website and engaging with new findings as they emerged. He was consistent in one thing throughout all of it. He presented evidence. He drew no religious conclusions. He invited scrutiny. That discipline, maintained over nearly half a century, is what gives his voice particular weight when he speaks about the DNA findings that emerged in 2022 and 2023. The story of the shroud's DNA analysis begins in 2015, when Italian researchers extracted genetic material from dust samples collected during conservation handling of the cloth. Their findings were informative but inconclusive, revealing human genetic material alongside plant DNA, suggesting the cloth had been present in multiple geographic regions across its history. In 2022, a team of geneticists returned to those samples using sequencing technology that had not been available 7 years earlier. What they found buried in the human genetic data had not been detected in the original analysis. The Human Genome Project and its successors have created comprehensive maps of genetic variation across human populations worldwide. Scientists can identify with considerable accuracy the regional ancestry of an individual based on the specific combination of genetic markers present in their DNA. European populations carry characteristic marker combinations. Middle Eastern populations carry different ones. The patterns of genetic variation that exist today reflect thousands of years of migration, separation, and interbreeding among human groups. An individual whose genetic markers are inconsistent with any of those established patterns represents a genuine anomaly, one that requires explanation. The DNA extracted from the blood-stained areas of the Shroud contained markers consistent with ancient Middle Eastern populations, which is geographically consistent with a first-century origin in the Levant. But alongside those markers were genetic variants typically associated with populations that would not, based on everything we understand about ancient human migration, have contributed to the ancestry of a single individual from that region. The combination did not match any known population group, ancient or modern. Furthermore, some of the variants identified were archaic forms that have largely disappeared from the modern human gene pool, appearing in the data at frequencies inconsistent with standard models of human genetic history. When Schwortz was brought in to review the findings before publication in late 2023, he approached the data with the same skepticism he had applied to every other claim about the Shroud over the previous 4 decades. Ancient DNA analysis is notoriously prone to error. Contamination from handling can introduce genetic material from dozens or hundreds of individuals over the centuries. Degraded DNA can produce sequence reads that appear coherent, but are actually artifacts of the degradation process itself. These are legitimate scientific concerns, and Schwortz raised all of them with the research team. What he learned from the methodology review was that the team had anticipated these objections and had taken systematic steps to address them. They had used multiple controls. They had distinguished between DNA from the image areas and DNA from background regions of the cloth to separate original material from later contamination. They had run the analysis multiple times to verify reproducibility. The anomalous sequences were not appearing randomly. They were appearing consistently in samples associated with the blood-stained areas, and they were showing the same unusual profile across multiple independent runs. Random contamination does not produce consistent, reproducible patterns. Degradation artifacts do not produce patterns that look like a coherent but unusual genetic profile. The data, whatever it ultimately means, was real data. Schwortz has been clear about what he believes the DNA findings do and do not establish. They do not prove the Shroud is authentic. They do not confirm any religious narrative. What they do is add another layer to a profile of anomalies that has been building since 1978. The image cannot be explained by any known artistic or chemical process. The carbon dating, as conducted, was methodologically compromised. And now the genetic material associated with the blood stains does not match any population group in our existing records. 3 independent categories of evidence, each generating the same conclusion. We do not understand this object. The scientific community's response to the DNA announcement has been measured. Several geneticists acknowledge the data was genuinely unusual, while arguing that the sample size was too small and the contamination risks too significant to support firm conclusions. They called for new sampling under tighter controls and for independent replication before the findings could be treated as anything more than preliminary. These are reasonable scientific positions, and Schwortz has not disputed them. His own call has been consistent. More research, better access, independent verification. The questions the Shroud raises are too significant to be left perpetually unanswered due to insufficient funding or restricted access. What Schwortz finds himself saying at 78, after nearly half a century of investigation, is something more uncomfortable than either confirmation or dismissal. He is saying that the evidence is real, reproducible, and unexplained. He is saying that dismissing the Shroud as an obvious medieval forgery is no longer a defensible scientific position. Not because faith demands otherwise, but because the data demands otherwise. The image properties do not match forgery. The DNA does not match any known population. The formation mechanism has never been replicated. And the object continues, after more intensive scientific study than almost any comparable artifact in history, to produce results that challenge our assumptions. He is also saying something that is perhaps harder to hear than either a miraculous confirmation or a clean debunking. He is saying that some of what we think we know about ancient history, about human genetics, and about the limits of what is scientifically possible may be less settled than we have allowed ourselves to believe. The Shroud does not answer questions. It asks them. After 46 years, Barrie Schwortz is still listening. Whether the cloth housed in Turin is what the faithful have always believed it to be is a question that science may never be equipped to answer definitively. But what Schwortz has documented over the course of his career is that the Shroud is, at minimum, a genuine scientific mystery of the first order. An object that should not exist as it exists, that contains information it should not be able to contain, that carries within it a genetic signature that does not belong to any population we have mapped, and that continues, in study after study, decade after decade, to respond to our best scientific tools with the same quiet, stubborn refusal to be explained. That is where the evidence stands. And for Barrie Schwortz, a Jewish photographer who never intended to spend his life on this question, that is enough to keep working. Because the truth, whatever it turns out to be, is worth the pursuit.